Hi, we're going to learn how to do one of my favorite functions with the heat map, which is called anchoring. So let's launch it, and then I'll describe how to do it. Up here on the anchor icon, let's click on that, and let's select a genome. And I want to select the Brucella microti genome, isolated from mice. So while this is loading, let me describe what it does. It's going to reorder the protein families in the order that they occur on the microti genome. So it's going to go, Brucella have 2 chromosomes. It's going to start with the first chromosome in microti, and it's going to call on the first gene. So if I scroll this open, this particular gene, the plasmid replicon protein, is the first called protein in Brucella microti. This is the second, the third, the fourth, and so on. So it's reorienting the heat map view dependent on that. It's not messing with the order of the genomes, just the protein families. And right away you can see patterns start to emerge. Let's open this a bit. And you can see some genomic islands that are well known in the Brucella. This one is called the Ink P island. If I go down to microti, you can see a number of protein families that are here. And because this is in microti, I know that they're next to each other. And so this is a large block of genes that are missing. And so let's look at who's missing them. They're missing in the abortus genomes, it looks like, except for this one, happens to have a few of them. But the large block seems to be missing in all the abortus. And here's another group, and you notice that it's highlighting on the vertical axis of the genomes that I'm going over. This is the melitensis group. They're also missing this block. If I were to scroll down, you can see that it's missing in these Pinnipedialis group, which were isolated from seals. These particular 2 genomes, this one was isolated from a human. It was very odd Brucella one. And this one from Suis biovar 5. So one of the questions, or one of the things that often you have happen with a really nice visualization is I can see the data here. I can see what it is, but if I scroll over it, I can see what it's named, but I can't capture that data. How can I capture that? I mean, if I have to write down all of these PL FAMs, it's going to take me the rest of the day. Oh, I would hate to have to do that. But watch my cursor now. I'm drawing, you see that blue square? Drawn over that in the microti genome that matches that area. Okay, so I've drawn that there. And then it's going to open a pop-up window that asks me what I want to do with this data. I can download the heat map data for the entire heat map. I can download those proteins. I can show those proteins, or I can add those proteins to a group, or I can cancel it. I want to show what those proteins are. So let's click on show. And this gives me a list of the proteins that I captured from that heat map. Notice that I said we were walking down the genome of Brucella microti and the genes that were annotated here. This is the ID for this particular gene in Patrick that belongs to this local family and this global family. Notice the genome ID here. Remember at one point we talked about unique identifiers in Patrick for particular genomes. That identifier is here. This is telling me that this gene belongs to Brucella microti. And then it's got a PEG number, which is just our identifier for a particular gene. And this is 349. This is the 349th gene in that genome. 50, 51, 52, 53, 54, all the way down to 65. These correspond to this area in this genome. And that is a really cool functionality. Next time, I'm going to show you how we can drill into specific genes and find out more information, not only about the gene from this table, but also looking at a gene neighborhood and figuring out how tightly conserved it is. Thanks for watching, and thank you for using Patrick. Bye.